Okay, so we're going to go over mitosis and meiosis, which both have to do with cell division, and one has to do with reproduction, the other one has to do with just how cells divide. Now, bacteria divide by binary fission, which isn't mitosis or meiosis, but it is a similar process. And if you remember that prokaryotes, or bacteria are prokaryotes, and they have a circular chromosome, and they only have one. And so before bacteria can split into two, it first replicates its DNA. And this begins at an origin of replication, so that N is cut off there, and then proceeds in both directions until you have two new strands of DNA, two new chromosomes. Then they go to the opposite ends of the cell, and septum forms and divides the cell into two different cells. Okay, septation then is the process by which a septum is formed, and the proteins involved in making the septum are called FTSZ proteins. So they make a ring around the middle of the bacteria, they cut it off and divide it into two. Okay, eukaryotic chromosomes, if you remember, are linear in shape, and there are more than one in eukaryotes. Most prokaryotes only has a single circular DNA molecule. Every species has a different number or arrangement of chromosomes. For example, chimpanzees and humans have the same number of chromosomes. All the chromosomes are pretty much the same size. It's just one of the chromosomes is flipped in chimpanzees compared to humans. So they have the same number of chromosomes, but the arrangement is different. Humans have 46 chromosomes. 23 of them are homologous or nearly identical. And if a person or if a fetus or developing embryo is missing a chromosome or has one too many chromosomes, then it usually is a fatal complication. And most stillborn births or early termination of pregnancies, natural ones, are because of a problem in the chromosome numbers. Alright, so what is a chromosome? A chromosome is a bunch of DNA that is packed together with proteins. And the term for that is called chromatin. So DNA and protein packed together is called chromatin. With heterochromatin, which is not expressed, it does have some value, so sometimes it's called junk DNA, but it doesn't make RNA, it doesn't make proteins, it's just kind of like filler DNA. Euchromatin does make proteins, or is coding for proteins, so it is expressed. And it's packed together very, very tightly. If you unwound it, it would, if you unwound a human chromosome, it would go from the moon, from here to the moon. And it's typically 140 million nucleotides long. Remember we talked about A, C, G, and T, those nucleotides. Or those are the base pairs of the nucleotides. Okay. A nucleosome, if you remember, chromatin is the whole structure. A nucleosome is in a smaller molecular structure, and that is a histone, which is a protein, and then DNA being wrapped around it. Nucleosomes interact with each other, and they create certain structures which make up the chromosome themselves. There are about 8 histone proteins for every 200 nucleotides, and the nucleotides wrap around the protein. They are positively charged, and they bond with the negatively charged phosphate groups of DNA. Okay. They, again, structure themselves into different structures called the solenoid, which is a bunch of these bound together and then coiled on top of each other. And this allows for the DNA to be compacted at the maximum. If it was all unraveled, then it would just be a big mess. So it needs to be compacted and organized. Okay. A karyotype then is a particular array of chromosomes, and this is a human karyotype we have a picture of here. And humans are diploid organisms, meaning they have 2 copies of each chromosome. Okay, 46 total, 1 from the mother and 1 from the father. Haploid means you only have 1 set of chromosomes. Some organisms are haploid. Our germ cells are haploid, which are reproductive cells. Okay, 2 chromosomes that are 1 from the mother, 1 from the father are called homologous, and individually 1 is called a homolog. So prior to cell division, the chromosomes need to be replicated. Okay, so if you look at this karyotype, you see that these chromosomes, this is actually 2 chromosomes, and they're in their singular form. When they replicate, then they look like these big X's. Okay, but these are both chromosomes. They just have a different amount of DNA. So once it's replicated, each chromosome has 2 identical DNA molecules called chromatids and held together by cohesion proteins. Okay, so typically when you look at a chromosome, or when you think of a chromosome, you think of these big X structures, but these singular chromosomes are chromosomes as well. So the cell cycle, how a cell is, every cell goes through different phases or different functionalities, and then towards the end of its cell cycle it will split into 2 different cells if it can. Some cells do not go through mitosis, but most cells do. Interphase is the working phase. That is where the processes are taking place that are important for the cell's function. So G1 is or gap phase 1 is the primary growth phase, the longest phase. S phase stands for synthesis, and that's where the DNA is replicated. And then G2 is gap phase 2, that's where it prepares to split and divide. Okay, and then M phase would be the mitotic phase, where mitosis happens. And it's subdivided into 5 other phases. And then the last phase, after everything is duplicated, you have 2 of everything, or twice as much of everything. Then you have cytokinesis, where a cell is split. Cyto means cell, kinesis means cut. So interphase, again, that's G1S and G2 phase. In G1, they undergo the major portion of growth. S phase, they replicate. In G2, the chromosomes coil, the centrioles replicate, and tubulin is synthesized. Okay, on our chromosome, you have a point of constriction called the centromere, and that's where it looks like they are connected. And they are connected by microtubules, and the site where those microtubules connect them is called the attachment site. Okay, in between them, we talked about our cohesion proteins. Then on either side, you have the connected core, and that's what attaches to microtubules. Each sister chromatid has a centromere, where it is thinner than the rest, and they attach by the cohesion proteins. Okay, again, there are 5 phases to mitosis: prophase, prometaphase, metaphase, anaphase, and telophase. Well, this is the figure from your book that breaks it all down into one big thing. We should be able to show you a picture, show you a figure of these are the nuclei. And you are able to then tell what phase it is in. In prophase, the chromosomes condense. So before prophase, when they're in their interphase, all the chromosomes are unwound and they are being used to make proteins. But then they need to be condensed into these linear structures. So that happens during prophase. And the spindle apparatus assembles. And this comes from centrioles, and this is in animal cells. It comes from centrioles, where the microtubules are being projected from the centrioles and they go to other sides of the cell. This circular array of microtubules is called an aster, and those are only found in animal cells. And then the nuclear envelope breaks down. So that needs to happen in order for chromosomes to move to the other side of the cell. The prometaphase starts when the nuclear envelope is dissipated. Then microtubules will attach to the different chromosomes, and each sister chromatid will be attached by the centriole on the opposite side. Okay? And the process of progression is where the chromosomes start to move to the center of the cell. And this is done, of course, again by the microtubules and the motor proteins that are at the connector part. So the motor proteins actually start moving the chromosomes. Metaphase is probably the simplest one to remember. Meta means meet. This is where the chromosomes align in the middle of the cell, okay, along the metaphase plate. And the metaphase plate isn't an actual part of the cell. It's just this imaginary line that is the middle of our cell. So they align towards the middle, and then anaphase, they start to separate, start to go to the opposite ends of the cell. And so in order to do that, the cohesion proteins need to dissipate, cohesion proteins that were aligning them, keeping them stuck together. Once that is removed, then they move to the opposite ends of the opposite ends of the cell. And there are two forms of movement. Anaphase A is the connector cores being pulled, so the actual microtubules pulling. And B, anaphase, is where the actual cell itself starts to move and elongate. Okay, so the poles, the ends, start to move apart. Telophase then is our last phase, and that's where the spindle apparatus disassembles, and you start to get a nuclear envelope enclosing the chromosomes again. They are completely separated from their other chromatids, and they are individually now chromosomes. Okay, the chromosomes then begin to uncoil and start to be used to make our nucleus. Okay, once you then have a nuclear envelope and the organelles and everything is pushed to opposite sides of the cell, you then have the process of cytokinesis. And there is a structure called the cleavage furrow in animal cells, and that's where proteins bind to each other and start to pinch off the cell. So this picture up here is a cleavage furrow. This has completed the steps of mitosis, and now it is now splitting into two separate cells. In plant cells, you have a structure called cell plate, which is actually a formation of the cell wall, which begins to separate the two sides of the cell. Fungi and some protists go through cell division a little bit differently. Their nucleus is actually separated. All the replication and mitotic events happen within the nucleus, and then when the cell divides, the nucleus divides with it, and the other parts of the chromosomes go with the other parts of the cell. Okay, so control of the cell cycle. How do these different chromosomes in different parts of the cell know what to do. Happens through a number of proteins, but there are two parts of the cell, or parts of the cell cycle, that are irreversible, meaning once the cell gets to that point, it will continue on to the next point. The first one's the replication of genetic material. So once that happens, you're going to go through mitosis. And the separation of sister chromatids. Once that happens, you're going to go through cell division. There are certain checkpoints at which the cell can be held. There's the G1S checkpoint, so that's the division between our G1 and S. And that's where the cell decides to divide. The G2M checkpoint, that's in between G2 and mitosis, and that assesses the success of DNA replication. If there was a problem with replication, then the cell will decide to either kill itself or not continue and just eventually die. And then there's the late metaphase, or the spindle checkpoint. Again, it checks to make sure the chromosomes have gone to the opposite sides of the cell. So you have these proteins called cyclin-dependent kinases, and these are enzymes that phosphorylate or activate proteins. And phosphorylate is the process of adding a phosphate group or an energy molecule to a different part of the amino acid or a protein. Okay, so we abbreviate these as CDKs, and they partner with these other proteins called cyclins. And cyclins are made at different points of the cell, and they regulate then the the different phases. CDKs can be activated or inactivated by phosphorylating different sites. So, for example, this one here, red would be, if that site is phosphorylated, then it is turned off. If in the green site here, if that site is phosphorylated, then it would be turned on. Okay, so then these complexes, they bind together, are also called mitosis promoting factor, or CDK cyclin complex is also called that. And then damage to the DNA will cause the phosphorylation to its inactive form, and mitosis is halted. So if there's a problem in replication, if the DNA is damaged, then the cell doesn't want to continue. You don't want to replicate damaged DNA because that can cause cancer or other problems. So we have two models, yeast and then our animal cell. In yeast, you have this G1S CDK, also called CDC2 G1 cyclin, and it increases the enzyme activity necessary for replication. So that would be this one right here, in between G1 and S. Then you have mitosis promoting factor. Okay, that's going to be right here at the G2M checkpoint, and it has CDC2 bound to mitotic cyclin. The same cyclin-dependent kinase is used, but it's bound to a different protein, different cyclin. And then the last one is anaphase promoting complex, and that will allow chromosomes to go through cytokinesis. Okay, that's in the yeast. The animal cells or multicellular prokaryotes are a little bit different. Rather than having one cyclin-dependent kinase, they have two. So it has CDK2, which binds to cyclin E at the G1S checkpoint. The mitosis promoting factor is done by a different CDK, CDK1, and it binds to cyclin B. Anaphase, though, is the same. The anaphase promoting complex is the same. Okay, so the eukaryotic one is just a little bit more complex because it uses two different CDKs rather than one. Okay, there are other proteins also involved in cell division. some are called growth factors, and they act by triggering our intracellular signaling system. So that means it's within the cell. It will bind to receptors and will cause the cell to grow, to replicate. The first one discovered was called platelet-derived growth factor, and what they can do is override the inhibitions to cell division within a cell. Cancer is unrestrained, uncontrolled growth of cells. So sometimes at those checkpoints, the proteins don't catch the errors in mitosis and allow the cell to divide, and that may lead to a continuation of cell division, which will cause a tumor. Okay. In cancer, there are two different kinds of genes that can cause cancer when they are mutated. Tumor suppressor genes, a lot of these have been identified, and also proto-oncogenes. So a tumor suppressor gene, P53, is one of these that is very important, plays a key role in the G1 checkpoint. And this is one that monitors integrity of DNA. So if the DNA is damaged, it will repair, the repair enzymes will be stimulated and may direct the cell to kill itself. And if that one is damaged, then it will cause the cells to continue to divide even when they are have errors, and that may cause cancer. Proto-oncogenes are genes that are normal but become mutated somehow and then will cause cancer. Some encode for receptors for growth factors, okay, and will prevent growth factors from causing mitosis. And only one copy of a proto-oncogene needs to undergo mutation for uncontrolled division to take place. So only one copy of that will cause cancer. A few of these have been defined for breast cancer and ovarian cancer, and they tried to be patented in the news recently. The Supreme Court said that no, you cannot patent a gene. And Angelina Jolie was recently in the news because she had these ovarian and breast cancer genes, and so she had her ovaries and breasts removed so that she would not be at risk for breast or ovarian cancer. Alright, now we're going to get into meiosis, which is very similar to mitosis, but basically goes through it twice. So the sexual life cycle of eukaryotic organisms involves meiosis and fertilization. Again, we said humans and most organisms are diploid cells, meaning they have two copies of every chromosome, except for our haploid cells, which are our gametes, or our reproductive cells, or our sperm and eggs. And they only have one set of chromosomes. So the offspring inherit one set of chromosomes from their mother and one from their father, and they become diploid, and that happens through the process of fertilization. Okay, sexually reproducing organisms will alternate between haploid and diploid stages, and for us, the haploid stage is very small, very short in humans, but in plants and other organisms, the haploid stage can be actually very long. The zygote is that fertilized egg, another name for the syngamy, or the synthesis of chromosomes from the mother and the father, male and female donors. Then, once they're fertilized, it will go through cell division, it will go through mitosis, and continue to go through controlled mitosis until the organism is sexually mature and can make haploid cells through the process of meiosis. Meiosis is also called reduction division because you're reducing the amount of chromosomes. Results in a daughter cells that contain one homologue from each chromosomal pair. Okay, like I said before, it's similar to mitosis, only it goes through it twice, goes through cell division twice. And when you this figure is really good, where you have mitosis, where you're making a copy and then splitting into two separate. In meiosis, you make a copy, you split, and then you split again, so that you have 4 haploid cells. Each phase, you have meiosis 1 and meiosis 2. They have prophase, metaphase, anaphase, and telophase. And if you remember the acronym PMAT, those should be easy to remember. And there's no DNA replication between meiotic divisions. So it goes through replication and then divides twice. It doesn't replicate in between. The second meiotic division does not reduce the number of chromosomes, but it splits them up by sister chromatids. Okay, meiotic cells have an interphase period where they can be arrested, similar to the G1S and G2 phases of mitosis. After interphase, the germline cells enter meiosis 1, goes through interphase, replicates, and then goes through meiosis 1. There are 2 processes that lead to genetic diversity. One of them is called crossing over, and that's where you have you have a duplicated chromosome. One, say the blue one's from the mother and this red one's from the father. Parts of the chromosomes will swap, and that's called crossing over. So now one of these chromosomes from the mother has a bit of piece from the father, and same for the one for the mother, or father has bits of pieces from the mother. The place where it's crossed over is called a chiasmata, and chiasmata means X. And that's that contact between those different parts of the DNA is maintained until you get to anaphase. So in prophase 1, just like in prophase in mitosis, the chromosomes coil together, they condense, the nuclear envelope dissipates, and the microtubules from the spindles form. Just like in mitosis, each chromosome is composed of 2 sister chromatids, and then the process of synapsis is where the homologous chromosomes stick together, they become very closely associated, and then crossing over occurs. When metaphase occurs, then they meet in the middle. Okay, so they line up at the metaphase plate. And the chiasmata, the terminal chiasmata at the end of the chromosomes, continue to hold and cross over. And then the microtubules drag them to the opposite ends of the poles. But they line up as not on top of each other, but as sister chromatids, or sister chromosomes. Okay, so one from the mother, one from the father, they line up on opposite sides of the cell. Whereas in mitosis, they would be lined up, all of them on top of each other. Okay, this allows for, again, genetic recombination to occur. So rather than all the mothers on one side and all the fathers on one side, you can have some of the mother and some of the father on each side, and it just happens randomly, and this is called independent assortment. Again, in anaphase, they separate, they start to be pulled to the opposite ends of the cell. The chiasmata break, okay, so they're no longer held together. Homologs are separated from each other, move to opposite ends of the pole, and then they remain attached to their centromeres. And now each pole has a haploid number of chromosomes with a homologous pair. Okay, and they are independently assorted then to opposite ends of the pole, and now you have different cells. So each cell that goes through this has some from the mother, some from the father, and then also each of those chromosomes have crossed over. So there's a lot of genetic recombination, genetic mixing. In telophase, you have the nuclear envelope reforming, and they are not identical, like I said before, and cytokinesis then occurs to separate them. It may be arrested in this phase for a while, but eventually you will have meiosis II occurring, and it goes through the same processes again. So this meiosis II resembles a mitotic division because rather than lining up side by side, they line up on top of each other. They meet in the middle, right? And then the chromosomes are pulled to opposite ends of the cell. So now you have haploid gametes with half the amount of genetic material that a normal cell has. So in the end, you start with one diploid cell that replicates, and you have 4 haploid cells that come from it. These develop directly into gametes in animals, but in plants, fungi, and other organisms, they will then divide again, go through mitosis, and you will get lots of gametes from that one. And that's why, like pollen, pollen and other seeds, you have tons of them, whereas in humans, you have tons of sperm and you have tons of gametes, but the process in plants and animals is more numerous. Sometimes they do not, the chromosomes do not separate like they should during meiosis, and that will result in either an excess or not enough chromosomes. And this is called an aneuploid gamete. And this is the most common cause of spontaneous abortion, like I mentioned before. Down syndrome is happens when someone has one too many of the 21st chromosome. So when we compare these two processes, there are 4 features that are distinct to meiosis. Synapsis and crossing over only occur in meiosis. The sister chromatids remain joined at their centromeres throughout meiosis I, and that does not happen in mitosis. Connecticores of sister chromatids attach to the same pole in meiosis I, and DNA replication is suppressed between meiosis I and meiosis II. Okay, and then these are just figures highlighting those same things, the differences between meiosis and mitosis. So again, when you look at metaphase here, the chromosomes are side by side. In mitosis, they line up on top of each other. And that what happens then is it separates, and now you have one copy of every chromosome in mitosis. In meiosis, though, they separate, and now you still have 2 copies of each, and it will go through a cell division again in meiosis II. Okay, and here's just meiosis II, showing again you have crossing over events, you have independent assortment, so that each of these haploids are different. Haploid cells are different from each other, and that is why we are not exact clones of our parents, in addition to being you get genetic information from different individuals. Alright, that's it for mitosis and meiosis.